Dear students, now we will go into the topic that is vascular changes in inflammation. One of the most important topic of pathophysiology, because your entire pathophysiology, wherever you study, whichever system you study, there is a relation with the inflammation. And inflammation, as you know, are of two types. One is acute inflammation, the other one is the chronic inflammation. Here we will be dealing with the vascular changes that are observed in during your acute inflammation, obviously. Now, what do you mean by vascular changes? Remember, anything that is related or written with vascular, it is related with the vessel or the blood, that is blood vessel and the blood. So what happens? The first thing is that the vascular changes that are observed in acute inflammation is, first is vasodilation, second is vascular leakage and edema, and the third is leukocyte emigration. Now what is this vasodilation? Now vasodilation means that if this is my blood vessel, I will show it to you. If this is the blood vessel, the normal size, the increase in diameter is the vasodilation. Now why this vasodilation is taking place? The vasodilation usually occur wherever there is injury. Suppose you are running and you fell down. What happens? You will say after a certain time your inflammation has taken place to that region, and what happens? There is redness of your feet, and you feel the feet is little warmer. Why it is so? Because of this vasodilation, the blood flow has increased, and blood flow has caused both warmth and the redness over there. The second step is vascular leakage. Now what is this leakage? Leak means something is coming out. That means my endothelial layer is always intact. Means our endothelial layer of the blood vessel is intact. Whenever there is an injury or inflammation, what happens is this endothelial layer breaks. Here you can see it, it is given, I have given it over here. The endothelial layer over here, the gap has increased. You know, all cells of my body is selectively permeable. Selectively permeable means that it will allow few substances to pass. And here what happens is my endothelial layer are intact. They allow very few substance to enter through them. Here what happens? The endothelial layer contracts. See, the size of the endothelial layer over here are same, except this one has contracted. So contraction means pulling. Small, it has become small. If I say it has become small, as a result the gap has increased and they allow the substance from the blood. If this is my lumen, this is the endothelial outside, then what will happen? The substance from inside the blood or the lumen moves out to the exterior. And who helps this? It is mainly the histamine, the bradykinin, leukotrienes. They play a major role in increasing the vascular leakage, or we call it as increased vascular permeability. The next is leukocyte activity. Now leukocyte activity has three more types of activity. One is margination and rolling, second is adhesion and transmigration, and third one is chemotaxis and activation. Now what is margination and rolling? Just see the diagram. Now this is my, here is the endothelial cell. Fine, endothelial layer is there. And this is the lumen. This is endothelium, this is the lumen. These are the leukocytes. Fine. Normally what happens? The leukocytes flow inside the blood vessel. Whenever there is injury, what happens is the leukocyte comes and settle over here on the endothelial layer, wherever injury has taken place. Now after this is called margination. Then what is rolling? See, here adhesion has taken place. If you check, the adhesion of the leukocytes have taken place. Adhesion means it got bound to the endothelial layer. Rolling means, suppose it has got bound here, it moves, it rolls like this. The ball rolls, it will roll like this way and will come near to the gap that is present. Now, adhesion takes place by certain proteins like ICAM, VCAM, LFA, and the rolling takes place by certain proteins called E-selectin, P-selectin, and L-selectin. The next step that we see is, here, sorry, one second. Now, the next step here is your transmigration. Now, previously, in the previous diagram, you had seen that it has rolled. Fine, this is the lumen. It has rolled and have come to the gap of the endothelial layer. What happens then? It moves out of the monocyte, the neutrophils. They move out of this gap and goes to the region of injury. And this is called diapetesis, and they usually is convinced who helps in this movement or diapetesis. It is PCAM, that is platelet endothelial cell adhesion molecule. Now the question may come to you that why suppose my neutrophil will go to the injured area? I am telling you, suppose you are coming to me. Why you will come to me? The question is like that. I should have some point, or I have called you, or something is with me, then only you will be coming to me. Suppose I have your marks, so you are coming for the marks, or I have the class notes, so you are coming for the class notes. Similarly, why this neutrophil or the monocyte will go to the injured area? Because there is chemotaxis. Chemotaxis means the chemical attract these cells. Whenever there is an injury, there are certain chemicals like cytokines, leukotriene, they get released into the injured area. And these chemicals, they attract the WBCs, that is monocyte neutrophils, they get attracted. Then once, what happens, the next step is, once they move, that is your WBC, once they move to the injured area, whatever foreign substance is present, they undergo phagocytosis. What is phagocytosis? That is, they engulf the foreign substance and they eat it up. And there is degranulation of the foreign substance. It can be any chemical, it can be any, this one, what I say is your microbiological substance, like it can be bacteria, it can be fungi, whatever is it, because of the main cause is the oxidative burst. This is the mechanism. Let me explain it to you. Now, this is my cellular layer. This is the blood vessel. Fine. This blood vessel, this inner layer is the endothelial layer. Suppose my cell or tissue got injured. Then what is happening? After injury, the bacteria is entering over here. Now, when the bacteria is entering over here, the bacteria have certain protein substance present on its present on its surface. This protein attracts the mast cells. That is, the mast cells are present below my cell surface or the tissue surface, and this protein gets attracted towards the mast cells. Once it joins the mast cells, what happens is, it releases the histamines. The histamine that is released, it has two functions. Immediately whenever the histamine will be released, two actions are seen. That is, one is vasodilatation and the second is increase in the vascular permeability. We have already discussed that what is vasodilatation and increase in the vascular permeability. Vasodilatation is that increase in diameter of the blood vessel. Dilate means increase in diameter of the blood vessel. What happens? This increase in diameter of the blood vessel then causes contraction of the endothelial layer. So gaps are formed. Here the increased gaps are formed. Now monocytes, neutrophils, RBCs, they are floating through the blood vessel. They come over here wherever gaps are there. There is adhesion of the molecule. After the adhesion of the molecule, they roll to the gap. After coming near the gap, they, by the process of diapetesis, they move out. Usually, which cells move out? It is not the RBC. It is actually for RBC, obviously it moves out, but for my activity, the pathological protection, it is the WBC that plays the major role, of which monocytes and the neutrophil plays the major role. Fine. Now what happens? These monocytes, after coming over here, actually what they form is, they form the macrophages. These macrophages, now they generate various substances like cytokines. Cytokines, the most common cytokines are the TNF alpha, interleukin 1, IL-1. TNF, the full form of TNF is tumor necrosis factor alpha. Now this tumor necrosis factor, or the cytokines, wherever they are released, they show two major activity. One is local effect and the other is systemic effect. The local effect is inflammation that you see immediately, and the second is your repair mechanism. That is, after injury there will be repair of your wound, and this repair mechanism is caused by your cytokines. The second is your systemic effect. Systemic effect means, that is the systemic system. What is the systemic effect related to blood? That is usually the the fever and the leukocytosis. What is leukocytosis? Deposition of the or increased amount of leukocytes. So these are observed later. So this is the total mechanism that is helping in the pain inside your inflamed area, or your inflammation is taking place, or this is the mechanism by which the body is fighting against the any foreign substance that is entering inside the body. So this is, hope it is clear to you, and I have tried to make it in a simple way. To know more about it or get means having an idea, there are the questions. Please solve them accordingly. You try to know what are they, what is their function, and as I always say, pathophysiology and physiology, please give diagrams. These are the references. Thank you, students.